Inheritance is the passing on of traits or characteristics from parents to their offspring. This is why when you look at a picture of your family, you will often see many similarities among individuals, but there will also be differences as well. So in this video, I will use genetic diagrams to explain the different ways that characteristics can be inherited from parents. So if you need help in this topic, keep watching. So before we go on to actually look at how characteristics are inherited, it's important that you understand some key genetic terms. So I have a list here in this table of some terms that you really need to know the meaning of. So first of all, you should understand what is meant by a gene. Is the basic unit of heredity, is a section of DNA which determines a specific characteristic or the protein to be made. So you would have understood this from the first video in Genetics 101 series. Now, the alleles now are closely connected to the gene. It's actually different forms of a particular gene that is responsible for determining the characteristics of the individual. So what you can do is think of the gene as perhaps ice cream and alleles as the different flavors of the ice cream. So you know there's strawberry, there's chocolate, there's vanilla. So think of alleles as that. Now, in terms of describing the different types of alleles, that brings us to the next two terms, the dominant allele and the recessive. So what do these mean? So the dominant allele is the one that is expressed more often and usually has the overriding effect when two different alleles exist. So a dominant allele is represented by a capital letter. The recessive allele, now on the other hand, that is only expressed if the dominant allele is absent. So it's really the dominant allele that has the power if it is combined with a recessive allele. So dominant overpowering, recessive is usually the one that would only be expressed as long as the dominant allele is absent, and that is the one that that's represented by the common letter. Now the next two terms, genotype and phenotype. So when we're talking about the genotype, that is the genetic makeup of the individual. So it's a combination of the alleles. So it may be a combination of two dominant alleles, a dominant and recessive allele, or two recessive alleles. So that is what genotype is. So the actual genes, the different alleles, the different combination of the alleles that the individual has. Now the phenotype is based on the genotype. So someone's genotype, the genes they have, determines the phenotype, which is actually the expression of the genotype. Phenotype will be the observable or the visible characteristics the individual has. So always look at it this way. Genes determine your characteristics. So genotype determines your phenotype. Now with the last two terms, homozygous and heterozygous. So homozygous is describing the type of genotype an individual has. So it's a genotype consisting of two identical alleles. So these two identical alleles, they can be either two recessive alleles or two dominant alleles. So these are the two identical alleles that are on homologous chromosomes, and I'll show you that shortly. Now with heterozygous, so heterozygous means different. So homo means same, so therefore you have two identical alleles. Hetero means different. So now you have a genotype with two different alleles. So one is dominant, one is recessive. So these are some key terms you need to know as we go on to look at the genetic crosses. Okay, let's look at this whole idea of the different genotypes that can be expressed. So first of all, remember genes, they exist in pairs at specific points which are loci on homologous chromosomes. Now, a gene is represented by two or more alleles using letter symbols. So we already established that. So I just have a table here showing you the three different genotypes based on the combination of alleles expressed on the homologous chromosomes of the individual. So what you're seeing here, so one paternal allele and one maternal allele. So I have it color coded. So the blue chromosome represents the one from the father, the pink one represents the one from the mother, and we're seeing the position in the location, the loci or the loci of the actual genes, the alleles that would determine the individual's characteristic. So in this first one, this individual has a genotype. So remember, genotype is a combination of the alleles. So we have two capital A's representing two dominant alleles. So this individual is described as homozygous dominant. Now, as it appears on the chromosomes, you can see that basically that is showing you that one chromosome, the one from the father, has a dominant allele at this position. Then the mother's chromosome has a dominant allele as well. So that is how an individual can become homozygous dominant for this particular gene. So they're carrying two dominant alleles, each from the mother and the father. Now in the second genotype, we have the heterozygous genotype. So you have a dominant and a recessive allele. So here we're seeing that the father's chromosome is carrying the dominant allele and the mother's chromosome is carrying the recessive allele. So this individual is heterozygous because they would have obtained a dominant allele from the father and a recessive allele from the mother. Now the final genotype, the final combination of alleles, is the homozygous recessive. So two common letters, two common A's there. So we have both father and mother chromosomes carrying the recessive allele. So this person is homozygous recessive. So you have to keep these three different genotypes in mind. So remember, the combination of these alleles is what's going to control and determine your specific characteristics. So let's look at this a little more closely now. So we have homologous chromosomes and the different gene positions, or the loci, as I talked about before. So we have the mother's chromosome and the father's chromosome shown here. So remember, this is a homologous chromosome. So if you recall from the first video in this series, a pair of homologous chromosomes, they carry the same sequence of genes, but obviously they would have different alleles. So as you can see here, the mother, so let's have a hypothetical situation. So if we have three genes shown here, P, A, and B. So let's just say P represents the skin color, A represents eye color, B represents hair color. So these are three different characteristics that are being controlled by these three different genes. But as you can see, there are going to be different forms of these genes, hence the whole idea of alleles. So you can see here in this case, the mother and the father, they have the same gene, the same allele for skin color. So we have two capital P's, two dominant alleles. So the individual is going to be homozygous dominant for this particular gene. So they're going to be homozygous dominant for whatever particular skin color. Now let's go on to the next gene, A, representing the eye color. So let's say, for instance, the mother has blue eyes and the father has blue eyes. So the allele A, the recessive allele A, is present in the mother's chromosome, the recessive allele A also present in the father's chromosome. So this individual, you would expect, would have blue eyes because it's inheriting both the recessive alleles from each parent. So they're going to be known as homozygous recessive for that particular trait. Now the last gene here, B, so that gene B, and you can see clearly that there are different forms on the mother and the father's chromosome. So this gene for hair color, so we have a dominant allele representing the one that the mother is contributing, and then the recessive allele the father contributes. So this individual inherits the dominant and recessive allele and therefore will be described as heterozygous. Their genotype will be described as heterozygous. So it simply is a combination of each of these three genes, the combination of the alleles, that would determine the individual's characteristics for skin color, eye color, and hair color. So you're seeing how the mother and the fathers, how their traits, how their characteristics, the combination of them would determine the actual phenotype of the individual. So we have the genotype determining the phenotype of the individual, the child. Alright, so hopefully you have an understanding of how the homologous chromosomes work in terms of the positioning of the genes. So we're just seeing three genes represented here, but in reality there's a whole range of genes that will be aligned along each of these chromosomes, but this is just highlighting three particular genes at specific points along the chromosomes, the homologous chromosome. Alright, let's move on to now look at monohybrid inheritance. So this is one of the first basics of genetic crosses, monohybrid inheritance, simply showing the cross between two parents with specific focus on one specific gene that will control one characteristic or trait. So this is the basics. There's also dihybrid inheritance and trihybrid inheritance, but we're looking at the basics in this video. So with monohybrid inheritance, the characteristic being studied in the genetic cross will be governed by two or more alleles for a single locus. So locus, remember, that's the position, the specific point of the genes on the chromosome. So we're looking at one gene. So just think of it that way. One gene, and you can have two or more alleles. So once again, we have the letter symbols representing the dominant alleles and the recessive alleles. So what I'm going to do, I'm going to show you how these genetic crosses can be conducted using two different diagrams. The line diagram method and then the more common Punnett square method, which is shown here. So let's begin to look at one type of way that characteristics can be inherited, and this is known as complete dominant. So this simply means that the dominant allele has the complete overriding effect when a recessive allele is present. And the example I'm going to look at is the inheritance of albinism, which is a recessive condition. So albinism is the condition in which pigmentation fails to develop in the skin or the hair. So you can clearly see, so this is a mother here with her daughter, and the daughter has no pigmentation, so she appears very pinkish, her hair is very light colored. So you can see the differences between the daughter and the mother. Let's look at how this condition could be inherited. So first you have to understand that there are two alleles that will control pigmentation in an individual. So we're going to let capital A represent the dominant allele for normal pigmentation. So having normal skin pigmentation, having the melanin being produced as usual, and the common letter A would represent the recessive allele for the lack of pigmentation. So this is the albino condition. So you're seeing that there are two different alleles which are controlling pigmentation in the individual. So it all depends on the combination of alleles that the individual receives that would determine if they have normal skin or albino condition. So note, a child will only be an albino if they inherit two recessive alleles from its parents. So that means that in order for someone to be an albino, they have to have the homozygous recessive genotype, which is two little a's. So let's look at the first diagram here using the line method. So what you're seeing is, so we have this genetic cross setup. We have a heterozygous parent mating with a homozygous recessive parent. So let's say that the mother is the one with the normal skin. So her phenotype is normal skin, and therefore her genotype, in this case as said earlier, would be heterozygous. So she has a dominant allele for normal skin color, so that has the overriding effect over the recessive allele. So she has two different alleles. She's heterozygous for normal skin color, so her phenotype is normal skin. Now the father in this case, he's albino because he's homozygous recessive, so always keep that in mind. In order to be an albino, you have to be homozygous recessive, so two little a's. So using the line diagram, the line method, so we're going to have the gametes. Remember the gametes, the sperm and the eggs, they usually would have half the amount. So remember, gametes are haploid, so they're not going to have the full complete set of chromosomes. So what you're seeing represented here, you're just going to have the dominant allele and the recessive allele from the mother, from her genotype, separated to just represent that, oh, this particular dominant allele would be found in one egg and the other would be found in another egg. So this is just representing the different types of alleles that can be found in the mother's eggs that are produced. Now, similarly with the father, well, he only has recessive alleles, so all of his sperm are going to produce recessive alleles. So you're just separating them to represent that these are the gametes. So now to determine the genotype of the offspring, the first generation, so you're simply just matching up. So you can see here that the mother's dominant allele matches with the father's recessive allele. So that gives us the genotype of the first individual. So big A, little a, so this first individual is heterozygous. So now for the second child, we're going to take the same dominant allele from the mother and match with the other recessive allele from the father. So you're pretty much just matching up each of the mother and father's alleles with each other. So for this second child, we have that dominant allele from the mother matching with the other recessive allele from the father. So that gives us child number 2, who also is heterozygous for normal skin color. Now in the third child, we're going to match up the mother's recessive allele with the father's recessive allele. So that is going to give us the third child being homozygous recessive, so 2 little a's. And then finally we have the mother's recessive allele combining with the father's recessive allele. So that gives us another homozygous recessive child. So basically in this scenario, in this particular genetic cross, you're seeing that a normal skin mother who is heterozygous for that normal skin, and a homozygous recessive father who is albino. Out of 4 children, you're going to produce the likelihood, the chances of, because you're really just working out the probability. So the probability of producing children that are going to have normal skin, you have 2 of them out of the 4. And then those that are going to have the albino condition, you have 2 albinos here. So it's pretty much like a 1-to-1 ratio. So you have 2 normal skin to 2 albinos. So that is just the probability of the children having normal skin versus being albino. So that is using the line method. So let's now look at the other method using the Punnett square, and this is usually the most preferred method. So we have our information set out, so which allele represents the normal skin pigmentation, which allele represents the albino condition. So the information we have, parental phenotype, so both of them have normal skin, and the genotypes, both of them are heterozygous for this normal skin. So they're one dominant allele, 1 recessive allele. So how we set up the Punnett square, so we're going to put the gametes for the father at the top as shown here. So the father has the heterozygous genotype, so he's carrying 1 dominant allele and 1 recessive allele. The mother is the same thing. She's carrying 1 dominant allele and 1 recessive allele. So it's just a matter of matching up each of the alleles, similar to with the line method, only thing the setup is different. So we're going to match up the mother's dominant allele first with the father's dominant allele. So that gives us the first child. So this child is homozygous dominant for normal skin. So normal skin is the phenotype. The 2 letters here, the dominant alleles represent the genotype. So that is the first child. So in the next case, the next child, we're matching up the mother's dominant allele with the father's recessive allele. So that is going to give us this second child. So this child is also going to be heterozygous, like the parents, like both parents, because it's obtaining a dominant allele and a recessive allele from each of the parents. So that would also produce normal skin. Remember, the dominant allele has the overriding effect over the recessive allele. Now for the third child, we're getting the recessive allele from the mother and the dominant allele from the father. Now usually how it is expressed when you're laying out the Punnett square, you must put the dominant allele before the recessive allele. So you may be wondering why I have the father's allele before the mother's allele, but normally you put the dominant allele before the recessive allele. So we have little a and big A. So we're going to put the A, the capital A, in front of the common A. So we have another heterozygous genotype there producing normal skin. So the final child is going to obtain a recessive allele from each parent. So 1 from the mother, 1 from the father. So that completes this Punnett square. So it's just simply matching up each of the alleles from each of the parents to produce the genotypes for each of the 4 children. So this is just giving you the probability for if they were to have 4 children. So basically the offspring phenotypic ratio would be 3 children having normal skin. So this one, the second one, and the third one. So these are the 3 children with normal skin. And then the last child here is the one with the albino condition. So that gives us a 3-to-1 ratio. So that is using the Punnett square method. Okay, so we're going to finish there with complete dominance, how we inherit certain characteristics through that type of inheritance. Now let's look at incomplete dominance. So this is a situation where you're going to have neither allele having the overriding effect over the other. So typically with the complete dominance, you have the dominant allele, the recessive allele. But in this case, when they combine, when they come together, neither one of them has the overriding effect. So we're going to look at the first example, which is flower color in a particular species of flowers known as impatiens. So allele R would produce red flowers. So when you have a red flower produce, that phenotype red, that is produced because of the genotype RR. So allele R produces red flowers. Now allele W will produce white flowers. So when you have two W's coming together, that would produce the phenotype which is white flowers. So when you have a combination of the R and the W alleles, that would produce like a mixture, which is a combination of the red and the white, which produces pink flowers. So as you can see that there's no complete dominance. There's no allele that is particularly dominant over the other. There's a combination of these two that would produce the express phenotype, which is pink. So whenever there's an R and a W, when those two alleles come together, they produce pink flowers. So this is the Punnett square here set up. So if we had two pink flowers, mating, so we have a genetic cross, two pink flowers, RW mating with RW. So we're going to do the normal matching up like what we would have done before in the other cross. So you're matching this R with this R to produce this flower here, which is the red flower because there are two R's. The next one would be an R and a W, which produces the pink flowers. You're matching up the R with this W to produce this offspring. So that's another RW. And then the last offspring would be the white flower, which is two W's. So the simple process, same process going on here. So as you can see, if you have two pink flowers combining, there's going to produce one red flower, two pink flowers, and that would be one white flower. So in this case, the phenotypic ratio is a little different. So we have one red to two pink to one white. So that would be our final phenotypic ratio. So basically for every two pink flowers that are produced, you would only get one red flower and one white flower being produced. So that is the first example for incomplete dominance. Now let's look at the next example, inheritance of sickle cell anemia. So in this case, we're going to use the letter A to represent the allele for normal hemoglobin and letter S to represent the allele for abnormal hemoglobin. So just a little background information about what sickle cell anemia is, the problem with this condition. So as you can see here, a sickle cell red blood cell has a strange shape, unlike the normal red blood cell, which is nice and biconcave in shape, nice and rounded. So what happens is that there's a faulty gene that causes the hemoglobin, which is the red pigment within the red blood cell. That hemoglobin molecule is going to be shaped a little different. So that's what causes the red blood cell to shape sickle rather than rounded. So the normal hemoglobin would give you the normal shape of the typical red blood cell, while the abnormal hemoglobin produces this sickle-shaped red blood cell. So that's just a little background about sickle cell anemia. So we have the letters here representing the alleles for the different types of hemoglobin produced. So basically, if the individual has two alleles, two A alleles, they have normal hemoglobin, they should have normal red blood cells. If they have two S alleles, they are going to be with the condition, they're going to inherit the actual sickle cell anemia condition. So now you need to understand now that if we have a heterozygous condition, so a combination of the A and the S, so for instance, so say the parents are heterozygous, so they're going to have the sickle cell trait. So this is the intermediate, the mixing of the traits, so both normal and abnormal hemoglobin. So pretty much they're going to have a combination of normal red blood cells and sickle-shaped red blood cells in their blood. So if the parents are both heterozygous, so they're they're known to carry the sickle cell trait. So they have the sickle cell trait, so they're heterozygous. Let's see what the Punnett square for them would produce in terms of the children, what the probability of their children inheriting normal red blood cells or the sickle-shaped red blood cells, or being a carrier or having the sickle cell trait. So as you can see here, the setup here, the Punnett square. So both parents are heterozygous. So one parent up here, we have A and we have S. The second parent, A and S. So the similar setup that we would have been doing all along. So in terms of matching up, we're going to have the A matching with the A. So that would produce the normal hemoglobin. So you would expect that this individual would have normal red blood cells. So when we're matching up the A and the S, that produces the sickle cell trait. So it's a combination, it's a mixture of having both normal red blood cells and sickle-shaped red blood cells. So the same thing here for this third child. And then the last child, this child would inherit both of the S alleles, both of the alleles representing the abnormal hemoglobin. So this is the individual with the sickle cell anemia condition. So let's look at the different phenotypes and genotypes of the offspring. So out of the 4 children produced, one child would have normal hemoglobin. So you would expect that their red blood cells would be normal because they're carrying both alleles that are producing normal hemoglobin. Now the next 2 children are going to be carrying the actual trait. They're going to have the sickle cell trait because it's a combination of the normal and the abnormal hemoglobin. And then the last child is the one with the sickle cell anemia condition. So once again, you're seeing that 1:2:1 ratio being produced. And just a little side note here, those that have the sickle cell trait, they usually have this resistance or protection from malaria, because the malaria plasmodium parasite, that usually doesn't survive well in sickle-shaped cells. So they have that advantage, that protection against malaria. Okay, so I'm going to wrap up here, and hopefully you have a better understanding of complete dominance and incomplete dominance and how these particular characteristics are inherited. So in the next video, I'm going to look at part 2 of inheritance of traits, and we're going to cover the inheritance of blood groups and sex-linked traits. If you found this video helpful, feel free to subscribe, like, and share. And don't forget to hit that notification bell.